And, okay, all right. So, mitosis in the cell cycle. You wrote a lot of this information down yesterday, and therefore you don't need to rewrite it. Let's say when you go back to study, when we do our test in another week, so about next week, Wednesday, we'll be having our test, all right? Because we're going to finish off this week. Monday we'll have one more lesson. Tuesday will be review, and then we will have our cells test on next week, Wednesday, okay? When you go back to study for that test, because we've done a lot of work online, you may be using your devices to read through some notes because we might not have the paper notes, right? So by all means, you can always go back and look at this or that handout that you completed yesterday. So we talked about how cells need to divide because we can't have these big, huge cells because the nucleus in the middle would never get any food. So when a cell gets so big, it undergoes this asexual reproduction where it simply splits into two. The splitting into two is called mitosis and cytokinesis, all right? Interphase. Interphase is a cell just living, doing its thing. It's not dividing, it's just being a cell. It's being a muscle cell or it's being a skin cell. It's simply existing as it does its job. When it gets to that proper size, then mitosis takes place. Remember, mitosis is the division of the nucleus. It's where the nuclear material divides to produce two nuclei in order to have the same atoms at the end. The nuclear material is the DNA. The DNA needs to divide and split so that we can have two identical nucleus at the end. Cytokinesis. Cytokinesis cuts the cytoplasm and makes two separate cells. It happens at the very end of the process. All right? Let's go through the stages of the process. And I'm just going to bring this up. And you would have wrote down little bits and pieces of this. But again, when we go back to study for the test, you can always go back and look at this as well. Interphase, it's the longest phase. It's the longest because it's just when the cells are just living. They're just doing their thing. Nothing special is happening. The cell carries out its regular life activities. In preparation for cell division, the DNA is duplicating. So we don't see it, but the DNA is making a copy of itself during this process. Once we get into the splitting phase, those copies then can go on to the opposite sides of the cell. So interphase, the cell is just living. You're probably going to want to take a picture of interphase of the 3 pictures that we take for our lab, because that's just a regular cell doing its thing. When you take the picture, you're going to make a label to the cell that's undergoing interphase. You might even have all 3 phases that I want you to take a picture of in one picture. That means you could actually reuse your picture 3 times and just point to the cells that are undergoing the different phases. All right? So you might be able to get all 3 of them in one picture and then just reuse the picture. So interphase, just a regular cell doing its thing. You see a nucleus, you'll see a cell membrane, a cell wall, etc., etc. So the cell division, the division of the cells themselves, 2 processes. One is the mitosis. That's where the stuff separates and goes to the opposite sides. And the second part is cytokinesis. That's when the cytoplasm splits and you end up with two separate atoms. Mitosis, the division of the nucleus and its content. Cytokinesis, the division of the rest of the cell and its cell membrane, etc., etc. So mitosis has 4 parts to it. And that's really kind of what we're looking at today. We say PMAT to help us memorize it. P stands for prophase, M stands for metaphase, A stands for anaphase, T stands for telophase. All right? Now, this information is what you wrote down, or similar to it, on that sheet that I gave you yesterday. This information is also found in the textbook. When you do the assignment that I'm going to give you in a moment, I'm asking you 2, describe what happens during the phase and draw a picture of it. So essentially, out of your textbook or using the note, you're going to summarize what's happening again, and you're going to draw a picture of it. You can use the picture drawing ability in order to do that, and you can use colors. We've got different colors to access there. In the little assignment, I'm asking you to draw with 4 chromosomes instead of 3 chromosomes that are pictured in your textbook pictures. This comes out of the textbook. So for prophase, you're going to sketch something like this. I don't need the interior of it colored unless you'd like to get really creative, but I do want you to draw the chromosomes and show me that the nucleus maybe is breaking down a little bit. The DNA condenses to form chromosomes. We see these structures that look almost like thick little strings, and we can start to see them. Each chromosome consists of 2 identical sister chromatids separated or held together by a centromere. So there's 2 parts to the chromosomes held together by this thing called the centromere. It's in the center. And the nuclear membrane is starting to disappear. It's starting to lose its shape. All right? So in prophase, we can see the chromosomes. They're still in the center. They're not organized in any way. And the nucleus is starting to break down. Metaphase. Meta means middle. So in metaphase, the DNA, the chromosomes, they line up in the middle. So you can tell that because they're all lined up in the middle. The chromosomes line up along the center of the cells. Spindle fibers, these fibers extend from the ends, and they grab a hold of that center, and they start to pull them apart. In metaphase, the pulling apart hasn't happened yet. They're all lined up, ready to go. So we have the chromosomes lined up in the middle during metaphase. Middle, meta. All right? Anaphase. Anaphase is where they start to move to opposite ends. I remember anaphase because of that starting of the word an. If you are anti-something, let's say you're an anti-environmentalist, you would be against the environment, right? Does that make sense? Anti means opposite. If you're anti-something, you're opposite to whatever that thing is. So in anaphase, think of it as moving to the opposite ends. You see how that's going to the opposite ends? So ana means opposite, like anti, and the sister chromatids are moving to opposite sides. They're getting ready to make their own nuclei on opposite sides. All right? So that's anaphase. So metaphase is in the middle, and when they start to move apart, anaphase is opposite. Telophase, and it's the end of this process. Telophase, we're getting ready. The new chromosomes stretch out to form long strands of DNA. In other words, you don't see those chromosomes anymore. They unravel, all right? You know how when you, I'm trying to think of an example. You know if you had like a string and you had maybe some weight tied to the string and you spun it around, how it curls itself up? You know what I'm talking about? I'm trying to think of an example where that happens, where a string curls on itself. That's what the DNA does. It curls up, and we can see it as this big fat curl. That's the chromosome. But in the end, it uncurls, and it becomes a big long string again, and you can't see it anymore. All right. The new chromosomes stretch out. In other words, they unspiral. They stretch out to form long strands of DNA again, and a new nucleus appears. That is really hard to find. Actually, that's not a bad one to find, because it looks like a funny cell that has 2 nuclei in it. It looks like a cell that has 2 nucleus in it, and that would make you go, hey, I think that's telophase. That's what's happening there. So you might be able to spot a telophase, all right? Now, once you've got the 2 nuclei, you need this cytokinesis. and that's where it begins to cut in half. All right, so it's still kind of stuck together, but it's cutting in half, and it's breaking apart to be its own cells. And that's given its own kind of name, cytokinesis. So mitosis describes the nucleus dividing, and cytokinesis describes the cytoplasm and the cell membrane actually breaking apart. Cytokinesis equally divides the rest of the cells into two new daughter cells. In plant cells, a plate forms between the two new ones to show that the new cell wall is happening. So you see this plate forming. Cytokinesis, a hard one to take a picture of. I'm guessing when you get your cells all lined up, you'll easily be able to see a metaphase because they're all lined up in the middle. You probably won't be able to see an anaphase, but you might see a telophase, which has then like a funny looking cell with two nuclei in it. And then the other one you can take is just an interphase, a regular cell behaving like it is. Okay. So here's a silly little joke. What did the biologist say when she dropped her microscope slide? She said, don't step on mitosis. Nothing? Not funny whatsoever? Just don't step on mitosis? All right, that's the end of that. Actually, here's a little animation.
In which the duplicated
Here, we'll listen to this.
separate to form two genetically identical daughter nuclei.
And then we'll do our thing.
The nuclear membrane disintegrates and the nucleolus disappears. As prophase continues, the chromosomes condense and begin to appear. Mitotic spindles, which are made of microtubules, begin to form between the poles, and kinetochores begin to mature and attach to the spindles. During metaphase, the chromosomes become attached to the mitotic spindles at the kinetochores and align along the metaphase plate at the equator of the cell. During anaphase, the kinetochore microtubules shorten, separating the chromosomes to opposite poles, while the polar microtubules and the cell elongate. During telophase, chromosomes reach the poles of the cell and begin to disappear. The polar microtubules continue to elongate. The nuclear membranes form, the nucleoli appear, and the chromosomes decondense. Telophase is followed by cytokinesis, during which a cleavage furrow forms in the center of the cell, and the cell divides.
Okay, so that's just a little something there. And again, that's the end of that. So right at this point, I'm going to ask you to grab a textbook, because at this point, you're going to use the textbook to do the first page of the assignment. Then when you're done that, right, and I get a chance to see that you're done, then you can go ahead and you can get a microscope, and we're going to look at the slides. So at this point, you need a textbook, and you can go to Google Classroom and you can find that page.